आज हम माइक्रोबायोलॉजी के प्रैक्टिकल पार्ट का टॉपिक देखेंगे, कल्चर मेथड्स। इन द क्लिनिकल लेबोरेटरी बैक्टीरियल कल्चर्स आर इंडिकेटेड टू isolate bacteria in pure culture from the clinical specimen are their identification by various tests determining antibiotic susceptibility, prepare antigens for serodiagnosis of infectious disease, maintain stock culture. Methods of culture: streak culture, lawn culture, straw culture, stab culture, pure plate culture, liquid culture. अब स्ट्रिक कल्चर देखेंगे, सरफेस प्लेटिंग. इट इज़ द रूटीन मेथड एम्प्लॉयड for bacterial isolation in pure culture. A platinum or nichrome bacterial inoculating loop of 2 to 4 mm internal diameter is used for making stray culture. This loop is first sterilized in the Bunsen flame by making it red hot and cooled by touching uninoculated part of the medium. Then a loopful of specimen is smeared onto the surface of a dried plate near the peripheral area. This is named as primary inoculum. From the primary inoculum, it is separated thinly over the plate by streaking with the loop in parallel lines. The loop is flamed and cooled in between the different set of streaks. It is done to obtain isolated colonies over the final series of streaks. The culture plate is incubated at 37 degrees Celsius for overnight. Confluent growth occurs at the primary inoculum and well separated colonies are obtained on the final streaks. Single isolated colony is the best to study the various properties of bacteria. अब लॉन कल्चर देखेंगे. लॉन कल्चर्स are obtained by flooding the surface of the plate with a liquid culture or suspension of the bacterium. Culture plate is kept for a minute and then excess material is poured off. Alternatively, the culture plate may be inoculated by a sterile swab soaked in liquid bacterial culture or suspension. Swabbing plate is then incubated at 37 degrees Celsius overnight to obtain bacterial colonies. अब यूजेस देखेंगे लॉन कल्चर की. लॉन कल्चर मेथड इज एम्प्लॉयड इन antibiotic sensitivity testing, disk diffusion method, bacteriophage typing. It may also be employed for preparation of bacterial antigens and vaccines where a large amount of bacterial growth is required. Stroke culture देखेंगे. Stroke culture is done in tubes containing agar slope and is employed for providing a pure growth of the bacterium for slide agglutination and other diagnostic tests such as urease test. Commonly used agar slope is nutrient agar slope. Stab culture. Stab culture is performed by a straight wire changed with cultured material bacteria by puncturing deep inside the agar. This technique is employed to demonstrate gelatin liquefaction, oxygen requirement of the bacterium by Hugh Lifson's oxidative fermentative OF test to maintain straw culture for preservation of bacteria. Pour plate culture. This is one of the best methods to determine the viable count of bacteria present per milliliter of a liquid medium or a specimen. The procedure is as follows. Tubes containing 15 ml of agar medium in each are melted and kept to cool in a water bath at 45 to 50 degrees Celsius. The inoculum to be tested is diluted in serial dilutions 1 is to 10, 1 is to 100, 1 is to 1000 and so on. 1 ml of each diluted inoculum is added to each tube of molten agar. Mix it well and the content of the tubes poured into a sterile petri dish and allowed to solidify. These plates are incubated at 37 degrees Celsius for overnight. Colonies will be seen throughout the depth of the medium and can be counted. Number of colonies are multiplied by dilution factor to know the exact number of bacteria. One colony is formed by one bacterium. Uses देखेंगे पुअर प्लेट कल्चर की to estimate viable bacterial count in a suspension to quantitate bacteria in urine cultures. अब देखेंगे liquid culture. Liquid culture in test tubes, screw capped bottles or flasks may be inoculated by touching with a charged loop or by adding the inoculum with pipettes or syringes. Bacterial growth is detected by observing the turbidity in liquid medium. This type of culture method is adopted where the number of bacteria in the inocula are expected to be small. Uses blood culture for and for sterility tests. अब लिक्विड कल्चर की एडवांटेज देखेंगे. लिक्विड कल्चर और preferable for specimens containing antibiotics and other antibacterial substances as these become ineffective by dilution in the medium. Liquid cultures are also preferred when large yields of bacteria are required. Disadvantage देखेंगे. It does not provide a poor culture from mixed inocula. Identification of bacteria is not possible.